step in synthesizing purine is we need to have a ribose sugar. Ribose sugar comes from the pentose phosphate pathway. Since the bond is small, every step will be removed, rubbed, so just take care of that. The whole pathway will not be able to present on the single board so that you get a comprehensive view. So each step will be written and it will be rubbed. Step begins with the synthesis of or the formation of a ribose sugar which comes from the pentose phosphate pathway. We know that as the part of oxidative phase of pentose phosphate pathway, we can get ribose 5-phosphate. Ribose 5-phosphate. Let us also pay attention to the structure. So ribose 5-phosphate, it will be like this. Ribose 5-phosphate. This is the alpha D ribose 5-phosphate. At the 5th position, there is a phosphate group. In the first step is now to make this molecule with the help of an ATP and energy-rich molecule. And that is an ATP is cleaved into an AMP. Once you cleave this into an AMP, what is left is the 2 phosphate groups. And that is a beta-gamma phosphodiester. That is what we call a pyrophosphate. That pyrophosphate is attached to carbon number 1. So this is carbon number 1, 2, 3, 4 and 5. That is attached to carbon number 1. If the structure will be like this. I am rubbing the AMP here. Maybe AMP is here. So this is the structure. Position number 1, a pyrophosphate is added. 2 phosphate groups. Now, there are 2 phosphate here, 1 phosphate here. This is called PRPP. PRPP stands for PRPP stands for phosphoribosal pyrophosphate. Phosphoribosal pyrophosphate, that is PRPP. An ATP molecule is involved in this synthesis. Therefore, if the name of the enzyme is PRPP synthetase. PRPP synthetase enzyme. This is a crucial enzyme. This enzyme has the capacity to regulate the synthesis of purines. Remember, it is based on the purines only we will synthesize, we will build the purine molecule. Unless this molecule, this substrate is not available, purine synthesis cannot occur. So how all these metabolic reactions are correlated and connected, that is the interesting aspect for us to note down. So once we have PRPP, that is this molecule, we begin in the next step. In the next step is to make a ring here. To make a ring, that is the second ring of purine, that is the imidazole ring. Let us construct the imidazole ring. In the construction of the imidazole ring, the next step is CH2OP. We have the PRPP, this is OP, OPO, PRPP. To that, what will happen is a glutamine is added. A glutamine comes into picture. Glutamine is added. We know that glutamine has got 2 amino groups. One of the amino group is given to this PRPP. The amide nitrogen is added to this, and once you remove that, it becomes glutamate, and that amino group is added to carbon number 1, and you have a product. Let me rub this portion. We have a product which will be like this. NH2 is added here. Amino group is added. And this becomes the N-glycosidic linkage which is connecting the sugar with the base. This is the N-glycosidic linkage, that is the beginning. An amino group is added. Now the name of this product, since there is an amino group, it is 5-phosphoribosylamine. 5-phosphoribosylamine is formed. Now this is catalyzed by an enzyme known as glutamine PRPP amidotransferase, a very important enzyme. Amidotransferase enzyme, another regulatory enzyme in the case of purine synthesis. Now, transferase, please also understand, it is a kind of a transamination reaction. An amino group is transferred from here, but this time an amino group is transferred not to a keto acid, but it is transferred to a sugar. So we have the first product which is formed, that is 5-phosphoribosylamine. Step number 2. This is now step number, the next step is step number 2 is once you have 5-phosphoribosylamine, we shall call it 5-phosphoribosylamine, PRA as a short form. The next step is to add a glycine molecule to it. So you know glycine, a very simple, very small amino acid. Once you add a glycine, you see what happens to this. So we will not be drawing this ribose structure now. So the ribose is here, and here is the NH2. This is the N-glycosidic linkage. This is the NH2 that has come from glutamine. Now to this, a glycine is added. So glycine, this is the carboxyl end. This is the alpha carbon atom, CH2, and this is the amino nitrogen. This is contributed by glycine. This product is known as 5-phosphoribosyl glycinamide. 5-phosphoribosyl glycinamide. Therefore, if the name of the enzyme is also 5-phosphoribosyl glycinamide, not writing the whole thing, 5-phosphoribosyl glycinamide synthetase. Why synthetase? Why synthetase? Because this addition requires the cleavage of an ADP to PI. Energy investment is a must in order to have this structure. So this is 5-phosphoribosyl glycinamide is formed. Let me give you a little clue. So we are here. We are here in this structure, and this is the N9. Look at this structure. We are trying to construct this imidazole ring. Therefore, this is going to be N9 which has come from glutamine. This is the whole glycine that is added. Carbon number 4, 5 and 7 are added here. So this is step number 2. Let us look at the third step. Once we have 5-phosphoribosyl glycinamide, now what is to be done is add N10 formal tetrahydrofolate. So N10 formal tetrahydrofolate will transfer the formyl group to 5'-phosphoribosyl glycinamide. We are trying to construct the ring. So what we are trying to do is, now we have reached up to here, we are trying to add this carbon atom. So the formyl group is transferred. So it is catalyzed by an enzyme known as transformylase. Transformylase, and this is GAR transformylase, and the product that we are going to get, we will write ribose 5-phosphate, to that the CHO group is added there. Okay, so this now the ring is not yet closed. Now this is the product that we are getting. This is this product is 5-phosphoribosyl formal glycinamide. So this is the product. This is the result of the third step. Now in the fourth step, what we are going to do is we want to link the ring and form an imidazole ring. But prior to formation of a ring, what is going to be done? At this position, an amino group is added. What will be the amino acid donor in this? It is going to be glutamine. Let us see, that is the fourth step. The fourth step begins from here, and the short form of this is FGAR. Now FGAR, add a glutamine, and you know that it becomes glutamate, and an amino group is added at this place. This becomes an amino group. And it requires the investment of an ATP molecule, ATP broken down to ADP and PI. That energy is utilized for the formation of this amino group, and it is a transamination reaction. It is catalyzed by FGAM synthesis. And the name of the product is 5-phosphoribosyl formyl glycinamide. An amino group, showing the presence of an amino group. This is reaction number 4. Once this FGAM, this is FGAM. Once FGAM is formed, the next step will be to close this ring. Now, in order to close the ring, it is very simple. Remove a molecule of water and the ring can be closed. In order to close the ring, an ATP is required, ADP plus PI. Closure of this ring occurs, a molecule of water is released, and you will have CN. An imidazole ring is formed. This will be in the ribose structure. An imidazole ring is formed, and this forms like this. Okay, H. Here it is NH2. NH. So this is the structure. Now you can see one ring has been, the imidazole ring has been formed. Now we are going to start the formation of the second ring. This is known as 5-phosphoribosyl amino imidazole. 5-phosphoribosyl amino imidazole. It is, the short form is AIR, and the name of the enzyme is AIR synthetase enzyme. Let us also look at this structure at the same time, and let us see how this whole structure is being developed. So right now this imidazole ring has been formed, and we are here. So let us number these ring and let us proceed. So this is number 9, this is number 8, number 7, and this is number 3, number 4, 5, these many are formed. In the next step, once we form AIR, in the next step is to add a CO2 molecule. Now, if the CO2 molecule is added and it is added to position number 5. So, in the addition of a CO2 is catalyzed by a carboxylase enzyme. So, when a CO2 is added, this is the structure we are going to get. So, ribulose 5-phosphate is here, and on this is the glycosidic linkage. NH2 is here, and a CO2 is added here. The CO2 is added. Now we will get a product that is 5-phosphoribosyl amino imidazole carboxylic acid. 5-phosphoribosyl amino imidazole carboxylic acid, that is called CARE. The product name is CARE. Now this is, therefore it's a carboxylase, it is AIR carboxylase enzyme. We know that carboxylases are enzymes that requires biotin, whereas this carboxylase is not a biotin dependent enzyme. So carboxylase added and we get this product, we get formation of a CARE molecule. Next step is step number 7. In step number 7, what we are going to do is we are going to add a molecule of aspartate to it. Now, in order to understand, let me put either COO here and clear this portion. A molecule of aspartate is added, that is in step number 7. Aspartate is added. We know that aspartate is an amino group donor. So when you add an aspartate, it requires ATP, which gets hydrolyzed to ADP plus PI molecule. And aspartate is getting added here. So let us draw the structure here. So this is ribulose 5-phosphate, and to that the nitrogen. The imidazole ring is formed, and this is the C double bond O. Here is the nitrogen, and in order to create more space, this we are removing this portion. Aspartate will be added here. So the nitrogen, the amino nitrogen, followed by the carboxyl carbon and the carboxyl group, then CH2COO. So the aspartate is added here. This product is called Cycle. Succinocarboxamide, 5-phosphoribosyl amino imidazole succinocarboxamide. And as we have seen, it is an ATP dependent enzyme, so therefore it is called Purine Synthesis enzyme. So once you have this product, that is step number 7, then you have step number 8. Step number 8, what is going to happen if you look at this structure? Step number 8 is, so we are here, we need this nitrogen, that is nitrogen number 1, that has to be retained. In order to retain, this molecule is removed. This molecule is nothing but fumarate. You would have encountered in the TCA cycle that this fumarate is removed. So therefore, step number 8 is to remove that as fumarate, and you have a product which is nothing but, this is the product. It becomes NH2. This product is formed. The enzyme which is catalyzing this reaction is succinylate lyase enzyme. Okay, Cycle lyase enzyme, which will release fumarate as one of the product, and you will have this. This product is known as ICAR, aminoimidazole, that is ICAR. Now, once you have ICAR, you look at the structure. You have created up to here. Now what is left is one C molecule, one carbon has to be inserted. That is step number 9. So in order to introduce a carbon here, in step number 9, what we are going to do is that carbon is contributed by the N10 4-mile tetrahydrofolate is added, which becomes a carbon donor. So tetrahydrofolate is released, 4-mile group is added here. Therefore, you are going to get the product like this, RP. Let me clear a little space. You have already noted down. RP is here. NH2 here, NH and a CHO is added. CHO is added here. Okay, so therefore this is happening with the help of N10 4-mile tetrahydrofolate. This results into formation of a product called FICAR and catalyzed by an enzyme transformylase. So let us rub all that is not needed anymore. What we have done is we have a product that is FICAR with the help of an enzyme transformylase. Now if you look at the structure now, the structure needs only a cyclic ring formation. Now this cyclic ring can be formed by removal of a molecule of water. Now once you remove this molecule of water, that is the 10th step, and the final step in the formation of inosine monophosphate, that is 10th step, a molecule of water is removed and you will get inosine monophosphate. Inosine monophosphate, the structure is going to be like this. A ribose 5-phosphate is here, then nitrogen in the imidazole ring. This is the structure of inosine monophosphate. Now, this is catalyzed. This particular step is catalyzed by an enzyme known as inosine monophosphate cyclohydrolase. Why cyclohydrolase? It is forming a cyclic structure. At the same time, it is removing a molecule of water. So the enzyme is inosine monophosphate cyclohydrolase is the enzyme. So you have this product. It is from this product we will create, or the cells will create AMP as well as GMP.